Good morning. Thank you very much for coming, and I've been looking forward to this meeting because I wanted to meet more of the people who are involved in coffee agriculture in Puerto Rico. I've been enjoying the development of this project with Jose Carlos and Paul Bayman. And my field of expertise, I'm a fungal geneticist. I've worked on fungal insect interactions for most of my professional career. And I used to work on fungi that were eaten by insects, and then when I went to the USDA, I reversed my perspective, and now I work on fungi that eat insects for a living. And my specialty is Beauveria, which is a sporulating mold. It's a, I get this right, it's a nice little white colored fungus. It's one of the most common insect pathogens that you'll find in nature. If you're in the forests and you look in moist areas and you find dead insects, frequently they're covered with a white mold, and that is more than likely Beauveria. Much of the work that I've done is at the genetic level. These things look very much the same, but actually harbor a phenomenal amount of genetic diversity within the group itself. And over the last 10 years, I've been involved in some aspects of their interaction with Broca, but this is the first time I've been involved in a sustained project, and I'm very excited about doing that. So my laboratory is in Beltsville, Maryland, just outside of Washington, D.C. And the nice thing about that is that I know the people on national programs, so it was relatively easy to develop this project once it got started. Okay, Beauveria bassiana is one of the first comprehensively described insect pathogens. It was described in the early 19th century by an Italian. He was a lawyer that really wanted to be a biologist, and he quit law and he studied Beauveria mainly because he was part of it was driven by the fact that they were a serious pathogen in the silkworm industry that they were developing in northern Italy. And also he was very intrigued by the fact that the fungus, when it attacks the insect, it will essentially mummify the insect. And one of his, he writes beautifully, and he wrote a long chapter about the possibility that one could use it to embalm people that you loved, and that you could preserve their bodies for posterity. But today we just recognize it for the very efficient insect killer that it is. Oops, sorry. One of the things in the laboratory, as I mentioned before, they're white. They all look very much the same, a little different, maybe a little yellowish color sometimes, maybe more sporous, but you really can't do much with the morphology. We like this fungus because it can grow on simple media, and you can produce it in high abundance. It's a very efficient way to produce lots of infected propagules. And currently we have 2, but only 1 commonly used registered strain for use in North American agriculture, which I think is too bad. They make the process of registration very difficult. So we only have 1. Each one requires many years, as well as considerable money in order to develop to pass all of the criteria that EPA puts out for us. More recently, we've discovered that Beauveria also has other lifestyles that it interacts with plants as an endophyte. It's also very common in the soil. We don't really understand its role in the soil, but literally if you were to go out and sample a cup of soil anywhere in any terrestrial habitat on the planet, even Antarctica, it's been isolated in Antarctica, you can usually find anywhere from 10 to over 1,000 colony-forming units from that small volume. So it's very common in the environment, which is a benefit for us. So I'm going to just give you the technical background on Broca that I came into this project, as well as what I've learned about Broca in Puerto Rico based on samples that we've accumulated over the last 5 years. So even though it was described of and is considered an asexual mold for the over 250 years since it was described, it turns out the modern phylogenetics have shown that they're actually associated with the sexual genus of Ascomyces, cordyceps. And here on the bottom you can see that is a beetle larva there, and they are obligate insect parasites. And this is where they food, off of the cadavers of insects. What Beauveria has discovered is a way to avoid sex, or at least minimize sex in its life cycle, but maximize its ability to produce asexually. So that's how we frequently see it manifested in nature as an asexual organism. So in my laboratory, the arc of my research over the last 12 years with Beauveria has proceeded from molecular systematics, which I'm just about done with, partly because I think I've done most of it, brought it up to date, and I'm getting tired of it. But really what I'm most interested in is the molecular ecology, how the diversity is distributed across the landscape, what the role of asexuality and sexuality are in its life history, and the population genetics and the development of molecular markers that people can use to access this information, and trying to integrate all of this information to do epidemiological studies of Beauveria. And coffee turns out to be an excellent system because Beauveria loves Broca. It's a very common pathogen on that insect. So this is just a summary of some of the phylogenetic work I've done with Beauveria, and it's not a very large group. There are about 12 species. Most of them are very relatively uncommon. Beauveria bassiana, which is the species that you see on Broca here, and is the most common species worldwide, is at this level of molecular sampling, doesn't show much diversity, and I'll go into a little more detail with other tools in just a minute. But although we find it mostly as a sexual form, you actually find it as this sort of yellow vertical stroma on which little sexual structures are produced. The group as a whole, identification has been a problem because of the simplicity and plasticity of the morphology. Indeed, there's a group, this species lineage here, Pseudobassiana, I described 5 years ago, looks exactly like Bassiana, and it actually can be a significant, in some types of pathosystems, as a pathogen of its own right of other types of insects. So it's really important to know what you're dealing with. So that we've developed various tools for relatively rapid identification to place these isolates into the various species limits. One of the things that I found after concluding that last study, I wasn't unhappy with the level of phylogenetic resolution within the Bassiana group. And so we started to focus on developing more variable markers, and we focused on regions in between genes which provide relatively concentrated packages of genetic variability. And I have to be honest, the first marker that I developed was an artifact of an experiment, but I followed up on it and discovered it was really variable, and it informed me that these energetic regions were very useful, but this is now really our core marker. Even though we now have genome sequences and we can presumably find comparable or even better markers for resolution, this one serves an extremely useful purpose in our laboratory for first-line identification. So we now have a total of 10 loci developed. We've sequenced over 3,000 strains from all around the globe, although we've concentrated in certain areas based on problems on Broca, Western United States for some other projects, and Africa, etc. So our sampling isn't totally even across the globe, but we've sampled all the continents pretty effectively so far. So this is just a summary phylogeny based on our 10 marker system, and you can see just by looking at the branches, there were, in the earlier phylogeny, we had sort of a flat backbone within the Beauveria bassiana clade. We now can resolve quite a bit of distinct terminal phylogenetic diversity across the group. And what we find are 3 really prominent groups. In most habitats that you'll go to, you'll find that one of these groups predominates in the community, but we also find other rare lineages. So Beauveria occurs in these multi-species communities, frequently dominated by a single species. And these species have slightly different geographic distributions, their concentrations, What is remarkable about Beauveria, even the rare lineages, you can find them on multiple continents. So historically, these fungi have been able to disperse very broadly around the planet. So there's an old hypothesis about microbes that everything is everywhere, and in this case, Beauveria, it's quite true. Everything is there, everything is everywhere, mostly, but oftentimes in very different abundances. So in Asia, there's a dominant group, A1, that goes all the way to Europe, which is probably comprises 70–80% of the environmental isolates that you process. But we also find it in low abundance in Africa, South America, North America. A2 is a group that has a moderate presence in Asia, but is the dominant group in North and South America. And A3 is another group that's centered in Asia. Asia is considered by experts who study cordyceps, the sexual stages, as being the center of diversity for this entire group of insect pathogens, of many other species other than Beauveria. And Beauveria also seems to follow that same pattern. So we see these major lineages as being firmly established and very genetically diverse in Asia. When we look at the aggregate samples that we've analyzed from different parts of the world, we see that they do occur in quite a high level of diversity. So one of the things about Beauveria communities is that initially you have a very high species level diversity. So there's quite a bit of, that sort of one level of genetic diversity within the group are very sort of distinct, almost species level differences. And in further slides, I'll talk about genetic differences within one particular group. Also, this is the Mycology, turns out to be a relatively rarely recovered group, at least its closest relatives. And I've also recovered the Mycology strain from 3 continents now. So it does have, where it's been used, it does show some ability to persist over time. Now, one of the big questions about asexual fungi is, are they really asexual? It's sort of a theme in mycology, and this is one of those that I followed up with Beauveria. And ascomycetes have a relatively straightforward system of mate recognition and sexual development, and they have what they call these MAT genes that control mating. And they're a very simple structure. They're of one of 2 types, either MAT1 or MAT2, and these are defined that ascomycetes are haploid. So at the mating locus, an individual strain will be either one MAT type or another. And these genes are characterized by a high level of sequence and amino acid similarity. So they are functionally different, but they're both transcriptional factors that regulate cell-cell recognition, sexual and regulate the induction of sexual development. So typically in a mating, you'll get 2 strains of different mating type. They'll fertilize and form a binucleate or heterokaryotic state for a very brief period of time. These proteins interact and then turn on the pathway for sexual development. Now, some species show, literally, not Beauveria, but other species of cordyceps show a pattern of annual sexual cycles. Beauveria probably reproduces on a scale of decades, hundreds of years, or even longer. It's rarely seen. I think there are only a half a dozen actual physical collections of Beauveria from nature of the species that are typically used for biological control. So sex is rare, and so that has certain advantages and alleviate certain concerns about regulating these organisms, because you don't have to worry so much about gene flow from your biocontrol strains into your populations of indigenous fungi. And there are many ways you can manipulate this or use this information to ensure that that doesn't happen. So within Beauveria bassiana, I characterized the mating system. This was sort of pre-genomics, but it has a fairly standard structure. It has a couple of genes that are characteristically border the mating locus, and it has the typical types of genes that you find in the mat locus, and we have a PCR assay, which is a very convenient way to quickly go through and assess the balance of the mating genes in the populations that you're studying. And then finally, one slide didn't quite translate there. We developed microsatellite markers for doing very fine-scale genetic investigations of Beauveria, and we have 19 of them. And so what they are short, repeated DNA sequences, and they evolve very rapidly and change in length. So we just amplify the region, and we can run it out on a high-resolution sizing DNA analyzer and detect these mobility differences between them. This is when I started. So if any of you have done population genetic works, you know that this was back maybe in the early 2000s. Now we have capillary electrophoresis with fluorescent dyes, and we can do this all very quickly in a much more sophisticated and precise pattern, analyzing the fragments in an automated setting. So we use these for strain-specific genotyping, so we could tell the mycontrol strain from indigenous strains in a habitat, and also the population genetic analysis. So the next few slides, I'm just going to go through and give you a background on information that I've developed for the pathogens that we've been collecting and analyzing from Broca here in Puerto Rico. So what we find, wherever coffee is grown and you have Broca, Beauveria bassiana is probably the most conspicuous pathogen of this insect. It really likes Broca. That's the good news. And it's also, as a mycologist, well, anybody who's a biologist understands when they work with an organism, you need to learn particular aspects about the organism's behavior and occurrence in order to go out and efficiently collect it. And Beauveria can be tough. Looking for dead insects is not a lot of fun if you're down in the forest litter, especially if you're in a place that has a lot of biting insects and things. It's not much fun, but Broca is a pleasure because it's pure white. First, you can see the scars or the holes where the insects bore in, but when the insects die, the adult insects, they oftentimes will come out to the edge of the hole. And they die, and the fungus grows out of the insect and produces a lot of spores. So you get this beautiful contrast of this little white tuft of mycelium and spores, and it's very easy to see against the contrasting green or red background of the coffee cherry. So literally, we just go out and pluck one off, put it in a packet. It's the easiest collecting I've ever done. And in the laboratory, you can see they sporulate very efficiently on the insects, and each of these little spheres here is probably an aggregation of several hundred spores. So even a small Broca can produce tens of thousands of spores. So they're a very efficient mechanism for amplifying that genotype in the environment. So if they're successful in infecting an insect and they can produce a lot of spores, it's quite, it certainly explains why you see it at such a high level naturally in the environment. So since the first time I came here, we found Broca, and every time I go out in the field, we always find a certain amount. So these epizootics are continuous and they're sustained, and now, based on information from Paul and Jose Carlos's group, is that they're also, the Broca is also on alternate hosts in the area. So there's a fair amount of resource for Beauveria to eat on. So I'm using this is a great opportunity for me, a biologist of this organism, to try to understand the nature of an interaction of this pathogen with Broca. Typically, Beauveria is a generalist pathogen in natural environments. They seem to attack multiple species of insects at the same time, so there is not very strong pressure of Beauveria on a particular insect. It's rather diffuse in the environment. This one's relatively unusual in as much as that there seems to be a very focused and continuous attack of Beauveria on this population. And hopefully we can use this information eventually to improve our control of the coffee berry borer. these are the, this is one of Jose Carlos's maps. Actually, it might be one of those maps. But these are the areas that have been sampled, and the sampling has been collected not in just when the students go out, if they see Broca, they put them in a pack and they send them up to me in Beltsville, and I've been collecting them. And this year I've been doing a lot of genetic characterization of them. So we have over 220 strains now collected over 4 seasons at multiple sites. And hopefully with this background information, it will inform us to refine our sampling strategy as we shift from a broad-scale survey of the Broca and the environmental parameters that it occurs in, to develop sort of more defined strategies at individual sites where we can actually understand more detail about the interaction. So in Puerto Rico, all of the infections that I've analyzed so far, all 220, well, except for maybe 8 or 9, are all in this A2 group, which is the dominant pathogen, the dominant Beauveria in North and South America, which is very interesting. Why we're not finding any other species on it, I don't know. I think it's a very interesting thing, but this mirrors at a more focused geographic scale exactly the results that we had when we did a broad-scale survey in 2006, where we looked at isolates all the way from Mexico down through Brazil and Western Africa. It was all A2. So this group, A2, seems to have a special affinity for Broca. And that's a very interesting observation, and it must hopefully contain some important information that we can leverage to our advantage. Based on our sequencing system, there were 8 different sequence variants. They weren't that divergent, but there's clearly variation within the population. And as an aside, we did see Mycology. Unfortunately, I don't have we didn't have really good information on where Mycology has been used and for how long, and whether our sampling was, how soon our sampling occurred after Mycology had been applied. But the fact that we only find it at a very low level would suggest that it's not establishing, it's not persisting very long after applications, at least on an annual basis, and it probably isn't dispersing very far from its point of application. In the analysis of the mating type, we find that both mating types are present. So, in principle, sex is potentially occurring in Puerto Rico in these populations, but we have a lot more data to analyze. And this is Omar, who does our videography and is putting together the informational background for us. This is he was surprised that these fungi were having sex out in possibly having sex all around us at some coffee farm. Now, in the microsatellite diversity, we found, looking at a selected sample of 42 individuals representing the different chronological, geographical, and the initial sequence typing diversity that we had, we found a tremendous amount of microsatellite polymorphism. Within this limited sample, we had 147 alleles distributed across 9 loci. That's a lot of information, with variation of fragment sizes from 3 to 15 per locus. Within that, there were 14 unique multi-locus genotypes. There's a process called clone correction where you try to identify individuals that are really the same clone but have accumulated some genetic differences. And I suspect that actually the actual number of genetic individuals is going to reduce down to less than 10, but that's going to take more information and more analysis. And one of the signatures of clonal evolution was the high level of unique alleles that you only found in one or a very small group of clearly related strains. So there does appear to be clones within A2 appear to persist for a long period of time, to the extent that they actually accumulate significant genetic differences from one another. So despite the circumstantial evidence for the potential for sex, we I believe that really the populations that are out there are existing as a clonal assemblage of genotypes that show a lot of superficial genetic diversity, but probably there aren't that many original genotypes that define that population. And that's going to be one of, at least from my standpoint, one of the interesting puzzles to figure out. So, just to summarize, A2 is the dominant pathogen of Broca in Puerto Rico. So here it is as an indigenous fungus that is naturally dominating as one of the dominant pathogens of Broca. And I believe there's a theory of integrated pest management called conservation biocontrol, where you try to manipulate the environment to favor the pests or the pathogens that you're interested in. And I think that this offers a good opportunity to work with that paradigm. We see quite a bit of diversity among genotypes, so it's not just one clone that has a particular superiority on Broca. It's actually a number of different genotypes. And the Mycology, which is our one sort of legal strain that we can use, really doesn't show to have a very strong persistence in the environment. That is seen as an advantage by EPA because you don't want to disrupt the natural balance of the indigenous species. But on the other hand, what it means is that you have to apply it more often, and it may be a more expensive option. So the Hawaiian group that we're interacting with now, they're also interested in developing indigenous strains from their own island because they have such a problem with invasive species. So we're hoping that these two independent cases, we can see how indigenous populations of insect pathogens manage to rise to the occasion with the introduction of this highly invasive pest. So in my research, my aspect of the research in this project, I'm interested in, I'll be doing much more extensive genetic analyses using even more sensitive tools, looking at the within clone diversity, looking at clonal divergence, working with the group in Puerto Rico, trying to identify functional differences amongst genotypes and seeing if we can select strains that are superior in their ability to attack and persist in the environments with Beauveria, with Broca. And then we would like to compare the performance of indigenous versus mycontrol strains to understand whether the strains here really offer particular advantages over mycontrol, and bring this together to develop an integrated pest management for this pest. So in closing, I'd just like to thank the people, former and current members of my lab that have worked in some way, mostly transferring or extracting DNA or helping in analyses on this project. And then of course, my two local cooperators and friends, Paul Bayman, Jose Carlos, and all their laboratories. And it's really been an exciting opportunity. So thank you very much, and we'll take any questions. Yes?
Is there any research addressing or looking for the connection, is there any, between the Broca genotypes and the fungi genotypes?
None that I know of, but when the problem first I became aware of it, I did a lot of reading about Broca, and apparently it has a very narrow genetic diversity. So
yes,
so there really aren't the opportunities for genotype interactions is relatively low. I mean, one might expect possibly that a particular fungal clone could be pre-adapted or just particularly virulent on Beauveria, but it seems that many different genotypes are equally virulent on Broca. So I think most of the strains that have been selected at Senecafe and these are oftentimes selected on the basis of how many spores they produce, so it's more efficient to produce them in mass quantity. I mean, that's a very important criterion if you're going to select a strain to produce for large-scale application.
And I wonder, is any interesting details or differences in the genotypes collected in the African region, in Ethiopia, the birthplace of the coffee plantation and the coffee tree, and also the coffee berry border?
Well, in the 2006 paper, I mean, what I was so surprised was to find that West Africa and South and Central America, it was the same group, A2. The African isolates had different sequence haplotypes, so we know that there were some genetic differences. There were genetic differences. They weren't exactly the same thing. But I had never, at that point, I didn't have the microsatellite markers, and I could not make the more fine-scale genotypic comparisons.
Very interesting. Your conclusions are very similar of what one of our scientists has been establishing in Puerto Rico just by observation. Dr. Fernando Gallardo has mentioned several times that probably Mycology is not as effective at what we was expecting in coffee form. And he has established that probably we have native Beauveria that have to be studied more in detail, and that is the robust information that you're presenting here, and that's why.
Yeah.
But my question is why if we have native Beauveria and you just start saying that it's everywhere and we can find it probably in every place, why we have the impression, what seems that the coffee berry border is going faster than Beauveria then? Why we have to apply more Beauveria for the control of coffee berry border if it's there? Why this is that we have the impression as farmers and technical personnel in the farm that probably Beauveria have not been as effective as we was expecting to?
Well, there are a lot of variables in the environment, but it's certainly an ecological principle that when you have prey and predator populations, they mirror each other, but they're out of phase in time. And I think that this is what is happening with the Broca and the Beauveria. The Broca gets started. The infections come in a little bit later, and so the population expansion of the Broca on an annual basis gets started earlier than the population of the Beauveria can respond to. So what integrated pest management tries to shift those curves so that they overlay so that you get more effective treatment early, because in almost any disease, any epizootic, if you want to break the cycle, the best place is the earlier in that phase as possible. And so I think that's what, environmentally, ecologically, that's what we need to do, is to try to figure out how to get the Beauveria active earlier in each cycle of food production and invasion by, each annual invasion by Broca. And there are also other things that are going on, trying to eliminate or control the off-season reservoirs of Broca in some of the native plants, possibly spraying, either cleaning, more effective sanitation, or finding ways to attack Broca that could be in residual fruits in the off-season. But you have to essentially crush that founding population down so that it allows the coffee to outrun the Broca. But I don't know if it will ever be possible to eradicate Broca. It doesn't seem, nobody's done it yet. It's just a question of control. Luz, did you have a question?
You mentioned in the Phylogeny tree that there is a global diversity between the isolates or between the races of Beauveria bassiana, that you maybe the distinguish between races, I think the 2A13, something like that.
Yeah, actually that was sort of a,
sort of that.
Because the tree actually is too big, I sort of squeezed it down and put some of the more interesting.
The question is, all these races that you identify, have you studied or have you observed difference between the pathogenicity or virulence between the races?
Well, nobody's really, a lot of people, I mean, there's the literature has hundreds, if not thousands, of bioassay studies that people have selected isolates from the environment towards different pests, but there's never been any genetic information that can relate to that diversity. So hopefully now that other people will start using that information to select isolates and compare. On the flip side of that, we now can sequence entire genomes, and I'm working with a scientist in Minnesota, Catherine Bushley, and we're sequencing 50 individuals from 15 of those groups. We're going to have complete genome sequences so that we can actually compare the genomes of these fungi to look for differences in genes and look for signatures of selection to see if we can actually see what types of genetic differences and capacities are developing amongst these different lineages.
But there have been identified these virulent genes?
Yes, I mean, there are proteases, there are various types of hydrolytic enzymes that are really good at digesting insects. They also produce a number of metabolites that are implicated in debilitating or killing the hosts. But what we really don't know, I mean, these are based on very limited studies of an isolate that somebody was, there's been no way to integrate this information in a genetic context. And so this is, I think, the really next exciting area of work that's going to be done with Beauveria, because we can now sequence every nucleotide in the genome, and we can really compare them in great detail. Yeah.
I wondered if there's, if you're looking into any impact of weather or climate and how that might affect sort of the balance between how prevalent the Beauveria is.
Well, that's one of the underlying questions that's motivated the first year surveying so many different farms at different elevations and different types of shade versus sun and mixed agriculture. It's my guess is that yes, they do have an impact, but there's also so much season-to-season variation that there's going to be a lot of fluctuation in these interactions that are being affected by the environment at any given location. Whether many observations in one year is going to be able to tease out factors or not, that's the big question I think that we're looking at. But that's one of the big problems is working with organisms. Living organisms as a control measure is that they are very susceptible to changes in their effectiveness based on environmental parameters. Yes,
Do you have information about Beauveria bassiana in twig border, Phylogeny compactus? Phylogeny compactus in twig border. This is another Broca in Phylogeny that affecting especially Beauveria canephora.
No, I haven't looked at any of those isolates yet, but that would be an interesting.
Ambrosia beetle is the name.
Yeah. And you find Beauveria on that? Typically, the ambrosia beetles are. You find Beauveria with the.
Yes, Beauveria is everywhere.
Yeah, they're pretty good. But usually, the ambrosia beetles, oftentimes, they have their own cohort of fungal symbionts.
Yeah.
So fusarium and other things, and they help regulate that microenvironment so that the ambrosia beetles are less susceptible to epizootics by some of the common antibiotics. At least that's my understanding. I have not specifically studied that.
Are there any estimations on how quickly those clonal variations originate? And I think it's probably difficult because dating populations of fungus maybe. That's
right. Right now, I'm just gathering the data, and I talk to anybody who has the impudence to use molecular data to actually try to date events. I am very interested in the longevity.